thank you very much for the kind invitation, and thanks for the introduction. Today I will be talking about spatial transcriptomics and the new methods that are coming up in this field. And I will give a few examples that how these spatial transcriptomics are being used in toxicology studies and for drug-induced injuries in order to identify biomarkers and mechanism of action. And I will also talk a little bit about the bioinformatic pipelines that are currently available, or like the gaps that exist in this area. Also, like really like in general, what are the key gaps that we need to address in order to make use of this technology for toxicogenomic-based risk assessment. Just a very brief sort of introduction, I guess. I know it's going to be redundant, but like I wanted to make sure that we are all on the same page about like what toxicogenomics is. What's the goal of toxicogenomics? It's really like we are trying to link toxicity with non-targeted molecular screening endpoints, such as gene, RNA, protein, metabolites, in order to combine these with toxicant-driven changes, which will give us the predictive potential for risk assessment. And the final goal is per se to make sure that we can use these sort of molecular biomarkers in order to reduce the reliance to animal testings and in the regulatory settings, and also reduce the time that is required for toxicology testing and studies to be completed. And this is being done sort of by designing screening assays and identifying biomarkers for adverse outcome pathways, for genetic toxicity testing, and biomarkers for different sorts of injuries and toxicities. And within the realm of toxicogenomics, transcriptomics is arguably the most widely used technology, which has been around for a really long time. And over time, people and organizations, they have developed really good workflows and identified reliable endpoints to be able to make use of this knowledge, and also like make this accessible and understandable for scientists and regulatory agencies in terms of mode of action of toxicity and like molecular initiating events for a specific toxicant. But still, I think that still there is a gap to make to transition from mechanistic understanding and mode of action to being accepted in internal decision-making processes, and also like regulatory decision-making processes. And I think like one way to address this challenge is to use novel technologies that are becoming available in the field of transcriptomics, in order to make sure that we can understand the mode of action of the toxicities, and also we can sort of like go to the like really detailed understanding of the biology of the system, so that we build enough confidence so it can be easier to translate this knowledge from the molecular perspective to decision making at different levels. When we look at the technologies that are around for transcriptomics, obviously, like the most widely used technology is bulk RNA, and which was done using microarrays, and also like more recently using bulk RNA-seq, which pretty much works in a way like you combine the entire tissue, all the cell types together, and you get like one single endpoint as a gene expression for that tissue type. But then there's single cell RNA-seq technology that became more popular, and I guess like now is the most widely used in current days, where we try to link cell types to the gene expression. But what you miss with this is really like the tissue itself, like where that single cell come from in the context of that organ, or like that system that you are studying. And spatial transcriptomics is sort of like trying to address that missing piece. Like, so we know what cells expressing what genes, but we don't know where they come from. So like by spatial transcriptomics, you are closing that sort of like that missing piece in that puzzle. So like that hopefully will give you a lot of insights in terms of understanding the cellular processes that is happening within that tissue. And spatial transcriptomics, I mean, it's already becoming game changer in biology and in medicine, because it's addressing this really like the ancient question of how the tissue organization relates to tissue function. And that's because that's really like where you are going to be able to identify targets for new drugs, and that you do it by understanding the disease biology, where you know like how individual cell within that tissue is going to impact the entire organ by depending on where that single cell is located. But we think like in addition to sort of like understanding the biology and drug discovery, there's great potential for this technology to be used in understanding the mode of action of toxicity. If we can develop the workflows that are sort of like standardized, and we can develop endpoints that are relevant for risk assessment. A few examples of why this spatial transcriptomics is becoming sort of like revolutionary in the transcriptomic field. I mean, that like for a long time there were technologies that could technically visualize gene expression in the tissue context. That's not new. But what is new about this technology is high-plex. Basically, you can measure many genes from the same tissue at the same time using these technologies. For example, the previous technologies were like single molecule fish or RNA-scope. You could do like 10, 12 genes per slide, but then you don't really get, and you need it to be like targeted. So like, you know what genes you are looking for. But with these technologies, in ideal scenario, you are technically going to get the entire transcriptome. So like, it's untargeted and hypothesis free. And this is essential when we are thinking of developing biomarkers, for example, or like discovering new biomarkers, because you don't really know what genes you are looking for. And I think that's the major sort of like the benefit or like the advantage of these technologies over previously known, especially resolved, like the gene expression models, or like the techniques that were visualizing genes on a tissue. In addition to that, you can, because you can also do like sort of like H&E imaging of the same tissue, you can start to link that gene expression to pathology, to toxicology, and you do get those informations exactly for the same tissue. In addition, you can, for the same tissue, you can use multiple slices of the same tissue and do multi-omics, for instance, like one for protein, the other one for metabolites, and things like that. So like that really like increases the throughput of what you can really learn from one single tissue. One benefit of this technology over single cell, obviously, like when single cell, you don't get the special resolution, but also like single cell RNA-seq is sometimes it's biased to cell type. For example, if you have adherent cells, like cells that clump together, it will be difficult to quantify their presence in your sample, because they're like, you are not measuring a single cell, but like it's clump of cells. In addition, RNA spatial transcriptomics is not destructive. So like you are not dissociating your sample. So like you are not impacting the RNA as much as you would do with the single cell. And in addition, you can also measure sort of like if you are measuring protein, you can measure the proteins even from like outside the cell, because as you are sort of like visualizing the entire tissue, not the cell types itself. And in addition, like these technologies, some of these technologies are available for frozen, fresh frozen, and also like FFPE samples. So it gives a lot of flexibility, especially like with archive samples. It's really essential. When we look into sort of like the evolution of these modern spatial transcriptomics techniques, we can categorize them into two major groups. The first one is image-based methods, and then like second one is sequence-based method. Within the image-based methods, one of the novel techniques called MERFISH, which is technically like as an expansion of single molecule fish, like in situ hybridization, but the technology, or like the trick is that they use combinatorial labeling, meaning like now you don't have like very few set of probes, but like you can generate so many different probes by like creating this combinatorial labeling strategy. And now like with that, you can do, instead of like one image, you can do like n cycles of imaging from the same tissue. By doing that, you can create multiplex. And basically, you can do more genes per slide compared to the single cell, single molecule fish that you were limited to almost like single digit number of genes. The second sort of like the method within image-based technologies is in situ sequencing. This is quite a new technology, and it's quite attractive in a way that it works. Basically, this method, unlike MERFISH, it doesn't use any probe. Instead, directly measures RNA and sequence basically RNA at the nucleotide level. But the way that it works is it converts RNA into cDNA amplicons, and manually using a confocal microscope, basically sequence that transcript. So here is just an example. You can see like basically nucleotide level information that are being generated using this technique. Although it's called in situ sequencing, is actually image-based because you are using confocal microscope to sequence the individual transcripts. And this is really like give you like in additional level of information, unlike MERFISH, because now like you are not getting like one single data point per transcript, but instead you are getting a sequence of that transcript explicitly. So you can do sort of like, you can identify SNPs, variants. You can, you can align that sequence into the genome of the species that you are interested in, and then you can look for novel isoforms that might be expressing within your tissue. That's going to be essential, like almost for every sort of work that's being done from drug discovery to toxicity studies, as we are learning more that novel isoforms are really crucial for in terms of like the way that they function and how they are translating into proteins. In terms, like, next, I wanted to talk a little bit about the sequence-based technologies. In this technology, the previous methods, you don't really need like the next generation sequencing. You don't, it's all imaging. But these technologies require both imaging and sequencing. The most famous or like widely used technology is for sequence-based method is called spatial transcriptomics. I mean, the name is a little weird, but it's like when it was generated, they called it spatial transcriptomics. But then, like commercial-based version of this method is called Visium. It's sort of like commercialized with this 10x Genomics vendor. So the way that it works is an array-based method, in a way that you have basically a slide. Each slide has like four arrays, and each array basically each array is like 6.5 millimeter, but within each array you have a spot. Each spot is around like 55 micrometer, and they contain a unique molecular identifier. And these unique molecular identifiers, what you will be using to track down like where that specific gene is being expressed. So like you would put your tissue on these arrays, and then like H&E stained image, and then like you would be doing microscopy where you can register the location of the tissue and the location of each of these spots that they have these UMIs, and then you would take that tissue and do sequencing, and after sequencing you would technically have like a sequencing information and imaging the information, and computationally bring back these two levels of information together and quantify the gene expression per single spot. And so technically you are going to get the spatial resolution level information. The challenge, I mean, this method is very widely used. The only problem is it's not a single cell level. So within that 55 micrometer spot, you can put, for example, like 1 to 10 cells. So it's not so you need additional computational analysis to sort of like use the convolution methods to convert that spot into cell type, which there are tools computationally available to do that. In terms of how widely each of these technologies are used, we do have like 10x Visium is obviously the like next generation barcoding is the one that is mostly used, and like many institutions own this instrument. But next to that, there's another sequence-based technology called Genomics from NanoString is also quite widely used. But the other technologies are really like taking up more recently, like MERFISH and in situ sequencing. But I would say looking at this sort of like exponential growth, it's clear that we are pretty much at the earliest stage of really like developing these sort of technologies, and they will start to, there will be more data sets generated, and I think that over time this dynamic is most likely to change. In terms of data analysis, again, there's quite a bit of a gap. Like the amount of data that is being produced, I think is more than what is being analyzed. So like you can see, like last year, almost half of the data sets that were produced were not analyzed. But also, I think there is still sort of like more work that needs to be done, and there are a lot of publications around data analysis of spatial transcriptomics data are being published every year. When we look at the how widespread these like data sets that are produced for spatial omics are in terms of species and in terms of specific tissue types that are sort of like analyzed, we can see that they're very much biased toward human and mice, and also like within these two species, basically like mostly like we have most studies are from brain, like, and very little data are still available for other organs. I think like we're like looking at this picture is like there is I would say like there is a big open space for almost like every sort of studies, and obviously like people first the first way that they apply this method is the early stage drug discovery and also understanding disease biology for good reason. They're like that's like where the new drugs will be sort of discovered. But I think like there is for toxicology is that there is really like a big open space in which like various species that are relevant for risk assessment could be studied, and also like the different organs that are relevant for toxicogenomics and biomarker discovery. Then that will, I think that they should be included. Around this topic, like there are very few studies around the sort of like a toxicity, toxicology in general. And I try to like give few examples that I could sort of like find, and I feel like they are relevant to go over them. But I want to mention like these are really like these examples are sort of like scratching the surface, and is really like the starting of this being they are sort of like the first works, but like really like there is a lot of work that needs to be done in this area. But the first example is where authors they validated a drug-induced kidney injury model. Actually, not drug. I think is a surgery injury model, and they could identify, basically like they use this 10x Visium technology, and they did map the gene expression over the course of injury and repair of the kidney. And doing that, they were able to identify some of the injury biomarkers that tend to lose their expression during the injury and then gain it back in the repair, or for injury markers where they show, first of all, like they show like region specific expression, and also they tend to express during injury and lose their function during the repair. For example, here one of the injury markers, LCN2, is shown that for a control mice, technically like that doesn't show any expression, but during basically repair phase, sort of like injury and repair phase, you see the highest expression, but it loses its expression again during the healing. But the opposite effect sort of like comes up with another biomarker, which seemed to show regional specific expression in the control mice. Well, however, it loses its expression in the injury and gains back during the healing. Another example that I wanted to go through is the drug-induced liver injury, and how authors use this spatial transcriptomics to sort of understand the biology of liver regeneration after injury. Liver is one of those organs that is essential to use spatial transcriptomics, but because of its biology. So like if you look at the liver and how cell types are distributed, there is a strong spatial patterns into spatial pattern and zonation in the cell type distribution, and also there are strong global gradients for oxygen and nutrients in terms of like how metabolites are distributed within liver, and that they're most likely to create sort of like these special patterns for gene expression. And in this study, authors use this especially resolved single cell data to be, and were able to show that various cell types, for example, proliferating hepatocyte, they transiently reprogram themselves during liver regeneration, and they sort of like they orchestrate the regeneration process after injury. Last example that I wanted to go through is sort of the is use of spatial transcriptomics in prostate cancer. One reason I wanted to show this example is really like the ability of the spatial transcriptomics in understanding not only like the tumor, like the gene expression within the tumor itself, but also addressing this long-standing challenge of how like the tumor is interacting with its associated neighboring environment. So sort of like in this study was like the whole prostate was analyzed by subsampling per se, and they were able to use 10x Visium technology, and I understand how cancer tissue interacts with inflamed region of the tissue and versus healthy region within prostate, and they were able to identify some of the key biomarkers. One of the like in general, one of the benefits of using this sort of a spatial transcriptomics is that you are going to get a lot of statistical power. So it's not that per tissue type you're not getting a single data point, but rather so many data points within, especially result data points, that does give you a lot of statistical power and reduces some of the noise that might be associated with bulk RNA-seq analysis. I wanted to also like talk a little bit of like, okay, so I mean, we introduced like 4 technologies for spatial transcriptomics, but sort of like the question is like, okay, what technology you would use and when to use them, and what are the considerations that you want to consider when choosing the technology. I think the major factor is still the cost. So like these technologies are quite expensive to perform. But in addition to cost, I think like you want to look into if when choosing the technology, you want to look into like if you want to do RNA only or RNA and protein together, since some technologies does allow you to do co-expression of RNA and protein. In addition, if you really need whole transcriptome or some targeted genes that you are looking for, so like that could impact the decision. But other factors like really like the resolution that is needed. If subcellular level resolution is really needed, or you would be okay with having like information at like a single cell or like few cell level. And in general, like if for whole transcriptome analysis, currently is a lot more feasible to do technologies such as sequence-based technologies such as Visium. But if you want to go to subcellular level, image-based technologies such as in situ sequencing, although like they will be having lower detection efficiencies, and that you might have limitations in terms of number of genes that you can analyze, but they might be better solutions if interested in doing subcellular resolution. Another aspect that I wanted to talk about is like, okay, so like what directions we think would be necessary to take in order to make these technologies applicable for toxicogenomics in general. I think like the databases are one of the major bottlenecks. The data are limited, but even those limited data are not accessible or as accessible as they should be. They are sort of like isolated islands within individual publications. Also, like linking the data to toxicity and pathology is another field that needs to be expanded and become more. There needs to be more data generated, focusing on risk assessment and toxicology specifically. And I think another major bottleneck is having scalable workflows. I think which is sort of like it's from both for like a wet lab to computational levels. So like in order to make sure that we can use these technologies and make them sort of like move beyond only like understanding a mechanism of action, but like making sure that this can be applied in regulatory settings, we need to optimize the workflows that are being used to analyze this, to generate the data, and also like to analyze the data. Also, the endpoints for these sort of data sets are not super clear at the moment. So like there are various computational tools that are coming up, and they each might actually do some different ways of normalizing the data, some different ways of sort of like measuring the spatial variability of the genes, which would make it difficult to integrate these data sets and also like report these data not in a single publication, but like collect as a collective. And also like the limitations in terms of like how making these data accessible and visually accessible for everyone, or like at least like interested stakeholders. When just very briefly about like sort of like some bioinformatic tools that are being used or to analyze the spatial transcriptomics data. I think the first thing is like really like understanding like what we want to visualize or like what we are going to report in terms of like the endpoint for these sort of data. I think like eventually, like what we want to quantify is like what genes show a spatial variability in a spatial transcriptomics data set. And this spatial variability could be at the tissue level, like let's say like you may also call it like low frequency patterns. Let's say when there are global gradients of metabolites from within a tissue type, and that may lead to sort of like a long range gradients in the gene expression within a specific, let's say like the tumor microenvironment. Or it could be at the single cell level and its neighboring cells. So like one cell interacting with its neighbor, like a cell-cell communication per se. And that's really like the sort of like high frequency patterns that needs different ways of, need different metrics to quantify those sort of interactions. So like understanding like what level of gene spatial variability we are interested in, and also like the metrics that we want to use is going to be critical in characterizing the spatial transcriptomics data. There are a few sort of like open source softwares available out there that can be used to analyze spatial transcriptomics. I think the most famous one could be ScriptBy and Gedo, but also like Surat, which is most commonly used for single cell RNA-seq, but like also they have functionality for spatial transcriptomics too, which they do provide the platform to integrate sequencing and imaging data, and also quantify especially variable genes and integrate across samples and data sets. But it's still sort of like evolving, and there are a lot more publications that are coming out every day, and one needs really like computational support, like full-time computational support, to really like understand the field and also like comprehend with the inflow of new computational tools that are being developed every day, and understanding what tools to use and when to use. And it's still really like not, I wouldn't say like a super standardized workflows. And some of the challenges that are currently are within the like data analysis of this sort of data is, I think the major challenge is like how to integrate imaging data to spatial transcriptomics data, from simply from cell segmentation methods to also integrating them in terms of some of the morphological features that are being extracted from image, or like the pathological features that are within the images, and how they can be integrated with this transcriptomics data. Also, like the cell deconvolution methods are still developing, and I think both in terms of developing machine learning tools, but also like they require more data to be generated. So like from various tissue types to be able to generalize these sort of like methods that identify the cell type using spatial transcriptomics data from these special profiles. In addition, like there are new mostly deep learning models that are being generated that really like try to identify the spatial variable genes from previous data sets that are generated. For example, like one of the interesting models that I find it quite useful is this graph convolutional network. It's a eSTAR GCN, which what it does is try to link morphological features from H&E images into spatial transcriptomics data and cluster the cluster the genes within the tissue context, not only based on their gene expression level, but also based on the similarity of those cell types that are within that tissue, and also like their similarity in terms of having pathological features. I think those sort of like integrated models that link pathology to gene expression and and also like morphology, cell morphology, cell types to gene expression, and classifying based on that are going to be critical in terms of understanding that like the disease pathology and also mode of action of toxicity, as it will help you to understand how single cell type is interacting by its neighboring environment. Last point is I wanted to bring out we do miss having sort of like public databases that are essential to centralize all these data sets that are being generated. Unlike what we have in macroarray or like bulk RNA-seq, that currently like now they are good public data sets that sort of like you can visualize gene expressions and be interactive with the database. Currently, there are very limited opportunities to do that with the spatial transcriptomics. There is one special database that's generated and sort of has like some data points for a number of species, but it's still very limited. And I think that there needs to be more done in that direction. And that's really like going to be, I think, essential when we look at the sort of like a survey data that shows where the transcriptomics data are being used within investigative safety and like for toxicity studies. Currently, we are using most of the data that are being produced are being used in understanding the mechanism of toxicity, and also in like it was one of the interesting factors was like there are a lot of data that are produced within pharma companies that are not really even analyzed. So I think like having sort of like these databases are going to be essential to make sure that we can, first of all, use the data that's available and also like move beyond understanding the mechanism and use this data for prediction. And just to summarize the existing gaps, I think I talked about like the lack of public databases that is really like I think going to be essential for the success of these new technologies. And on the other hand, like they are currently these techniques are not able to. They are only able to visualize mRNA, but you can't do non-coding RNAs or like microRNAs because they need this. The technology often needs this poly-A to be able to bind, and that's really like something that I think will be important for toxicogenomics studies. On the other hand, like more data needs to be produced for relevant species for risk assessment. But like currently, most of the data that are available are for human and mice. And I think also like the vendors that are offering spatial transcriptomics, they mostly, the reagents and the probes that they have mostly works for these two species, but it's more challenging to get data for other species that would be relevant for toxicology. On the other hand, like one factor is like really these spatial transcriptomics are costly and resource intensive, and that really requires, like within single organization, it requires collaboration and interaction across various departments, from toxicology, pathology, histology, computational support, and also like for like the data support in terms of like handling the data. So that's going to be essential if we want to make sure that like these data are produced in a timely manner, that in which they can be used for taking taking actions based on those data. On the other hand, because these technologies are resource intensive, I think like we need to work across industry to ensure that everyone we have standard pipelines from wet lab to dry lab to generating the data and analyzing. And I think that will reduce the time time and to analyze the data, and also like make it more efficient in terms of generating the data. And that would be the critical element in terms of making like moving toward like making this sort of like new endpoints applicable in regulatory settings. Last point, yeah, I think we see a great potential for spatial transcriptomics to use in investigating toxicogenomics-based risk assessment, but it's still, this field is pretty much under, even not, I would say like there are very few studies that explore these sort of endpoints, and I think like there is a lot more work that can be done in this area. And I think like the next big thing in terms of using this spatial transcriptomics is when we are moving toward multi-omics, when we are able to link these spatial transcriptomics data to the protein level information and metabolic level information from the same tissue type. And that's going to be essential to mechanistically understand really like what's the mode of action for toxicity, and also like predicting how the cellular processes work within that tissue type, and also this multicellular units. Yeah, thank you very much for your for listening, and if there are any questions, I am happy to take.
Great, thank you very much. If folks have questions, they can raise their hand or put them into the chat. I really like how you laid out some of the issues and what we need to move forward. You commented a lot on databases. What are some essential information that would need to be in a database for it to be useful to move the field forward?
I mean, one of the, so like the very basic, like data points, sort of like similar to what we have for bulk RNAs, like the tissue type, species, and also like the technology that used to produce these data. But I think like the major challenge is the endpoints. For example, like when we have bulk RNA, is easier, like you have one single expression level, or like you have like fold change and p-value, like looking at like treatment versus control. But here you have like sort of like entire tissue to visualize, and it's not super easy to integrate across samples. So either you need to like visualize all the tissues that are sort of like measured, or like find a way to integrate these samples together with some metrics, or some ways of visualizing it that becomes more interactive.
Great, thank you. I see Brian has his hand up. Brian, you should be able to unmute now.
Yep, there we go. Thank you. Ali, great talk. That's really excited about this technology, especially in terms of application to toxicology studies. Obviously, a lot of the focus with spatial transcriptomics is in vivo and using complex tissues to sort of find out mechanistically what's going on using this. In terms of sort of from a screening perspective, obviously we're trying to use something more human relevant, lower cost, a higher throughput, and a lot of that involves in vitro models. Where I see a sort of place for spatial transcriptomics are in more complex in vitro models, using organotypic type situations. Just sort of interested in your thoughts in integrating spatial transcriptomics with in vitro models, and also ideas in terms of increasing its throughput and lowering its cost to make it more available for dose dependent and time course studies that are important for those types of toxicological studies.
Yeah, I think one way that we were thinking, I don't know like how much I can share, but like we were thinking like ex vivo models. So like you can sort of like sample from like directly from like human, and then like do like ex vivo, like let's say like to take the tissue and add the compound and then visualize, basically like that, do the transcriptomics, not like in vivo, but like sort of like in the ex vivo models. And I think that could be one application for toxicogenomics, like sort of like a not really, like sort of like a middle ground, like not going like the cells, but like not really going in vivo, but like sort of like looking for ex vivo models and developing ex vivo models and see if they would be helpful. But in terms of cost, I think, yeah, I mean that's sort of like really like depends on like on vendor, and also like how much like in-house and like sort of like support there is. But I would say that's really like I think like big challenge for these sort of data sets, and that's where like I feel like I was trying to highlight that like the data sharing, like should be also like really like become, because it's difficult to generate these data, but when you generate, there's a lot of data that often like even like within publication, they are not really analyzing the entire data set. So like there's just like one subset of like the data set that is like few genes that is being analyzed. And so like I mean they like I would say like to encourage people like not to be selective in terms of what they are sharing in the publications. Often like these spatial transcriptomics, although like they have the entire data sets, they don't really share the entire data set in the public as a public resource. It's like sort of like I feel like that could be, if those data become more accessible, like there may be more analysis that can be done for those data.
Thank you.
Yeah.
Thanks, Brian. You talked about that a lot of what's done has been in mice and humans, and I was thinking we have a lot of issues already in toxicology of converting from, a mouse to a human. Does that spatial layer add even more of a issue with that? If we're looking at mice data and trying to make decisions for humans, maybe you could comment on how we would layer that information on there.
Yeah, I think, I mean, that's going to be a challenge. If we I think like for I feel like ideally we want to use like ex vivo models like for human samples, and that might be more like easy to translate. But I think that's the same challenge that we have with the bulk RNA-seq. There's like how much you can translate. But yeah, I would say that translation problem will still be an issue with the spatial transcriptomics. It's not. Yeah, it wouldn't go away.
Thank you. Deidre, did you have a point or a question?
Yeah, sorry, I can't seem to make my hand raise. So yeah, Ali, thanks so much. I think, based off of, Brian's question as well as, how do we, do, oh, sorry, let me see if I can put my video on. Yeah, there must be something wrong with my computer. Based off of how, the various different types of species, that we use across the, the industry, including, mouse, we do a lot of things. We do a lot of things in rats. And, the spatialomics, for mice, that is really what is mouse and human, like you said, it is really, difficult. I think the mouse is a much better space. Like when we're going on that, I think the other species, whether, you want to say cross, translational, when you get to monkey, when you get to dog, like Ali said, there's, it's really very limited. It is possible to do it, and that's what needs to really be developed in this area, because I think it has huge, big, advantages and really a lot of room for growth. Most people are doing it for, for pharmacology and, those earlier models, but I think that's where it's growing. That's what, at GSK, we're trying to expand that in that area, and really, trying to, pull that in, because we see the value in doing that. So I think that, across the industry, I think that's a big open space for us to, potentially explore, and how that data, gets integrated, across all of these various different endpoints. I think in regards to, most is done with tissues. And I think, Brian, to kind of go along with your point there, there are various different ways that, you can use frozen tissues, and you can use FFPE tissue, like Ali mentioned. But, there are, what I've done in the past in some instances where we wanted to look at, cells, we wanted to say microdissect out some cells, we really froze the, froze the cells in a suitable, medium, and you can make a block that has those cells in there. And then, that I would have to say could be suitable for, the spatialomic type, if you want to see like where in the cell that particularly is. I think it would be interesting, and I haven't really seen a paper out there yet about, organoids or anything like that, as to, where they would kind of apply this technique. Ali, I don't know if you know that's out there, but I personally haven't seen a paper. But anyway, I think it's a huge, big room for growth, across the industry. But thanks.
Thanks, Deidre. Ali, do you have any follow up to that, or can we go to the next one?
No, that's great. Yeah, like, I mean, like ex vivo models are like organized, and I think that they're going to be really like a big players.
Great. Bevan, I see your hand up. You should be able to unmute now.
Great, thanks. Great presentation. Thank you, Ali. We actually did some spatial transcriptomics on one of our mouse models, and it's looking pretty interesting. But one of the limitations is the amount of tissue that you're able to analyze is about 1 centimeter squared. And if you're a mouse, there can be quite a lot of interesting things happening in that small area. But if it's a rat, that's actually a very small amount of tissue that you can analyze. And then even more challenging, of course, would be human tissues, where the liver is enormous and you're looking at just this little tiny region. So I just bring that up as sort of one of the technical challenges with spatial transcriptomics.
Yeah, I think that's certainly like the limitations. I don't know like which technique you use, like most likely like 10x Visium, but I think like nanostring, like cosmics, they are now like Merfish, and now like they are actually like allowing you to go above like centimeter scale, like I have seen like, for example, like the entire mouse brain you can basically like visualize with Merfish, I think like few centimeter. And I think that those sort of like techniques could become useful. And also like if you think about it as like we can stitch the images together. So like, and like taking few, I think like more of a, I think histology is like sort of like the challenge there. But I think that some technology like nanostrings, I know like from like Vision, this Merfish company that does Merfish, they go beyond 1 centimeter. But I think like the next, I think like you don't expect like the 20 centimeter, like so it's just. But I feel like stitching images together will be another way of doing it. But I guess, yeah, I think that's sort of like one limitations for 10x Visium at least.